Ladies and gentlemen, thank you for coming to the late last session, and it's nice to see Ian Evett here because I met Ian about 30 years ago, something like that, and I remember having a discussion about likelihood ratios versus what I would call coincidence probabilities then, random match probability, and I was just arguing vehemently that they're the same, there's no difference. I am converted now, Ian, 30 years later. Anyway, today this is a joint presentation by myself and Mark Perlin, and it has to do with DNA mixture deconvolution. And it combines a couple of things. One is some technological advanced technology, I would say, using laser capture technology, and secondly, using quantitative computer interpretation. Hence, technologically oriented guy versus the quantitative computer gentleman there. So the title is DNA mixture deconvolution by binomial sampling of individual cells. Okay, so what I'm going to talk about is just have a brief introduction to mixtures, talk about the laser capture technology that's used briefly here in this work, then talk about the proof of principle experiment that we conducted, basically the experimental strategy, and then we'll talk about the quantitative computer interpretation that Mark will then take over from me at that point. Okay, it's well known in forensic casework, the forensic casework world, that there are different, when you have a mixture of DNA, there are different types of mixtures, and these different types of mixtures can either facilitate the deconvolution of that mixture or make it more difficult. And mixtures with major minor contributors are reasonably easy to deconvolute because you have a major contributor, and you can normally deduce what that major contributor is. The minor contributor, of course, may be more difficult because of allele sharing, etc. We also have mixtures with no contributors. That is where one of the contributors, the assumption is made about who that person is, and we can then subtract that profile from the overall profile. And I'm talking here in this talk only about two-person mixtures, okay? Since these are the most commonly found mixtures. And it's this third class of mixtures that I'm going to talk about here today, and these are the mixtures with indistinguishable contributors. That is the situation where you have very little, you know there's a mixture, you know it's, you deduce or assume it's a two-person mixture, but it's not easy to deconvolute that mixture because the band intensities are similar, etc. And this is particularly difficult when and most difficult when the mixture weight is 50-50. 50% of one contributor and 50% of the next contributor. Obviously, if that's the case, then it's going to be more difficult to actually deconvolute that mixture. Okay, so what typically happens in a mixture, is that here's a two-person mixture up here, blue and white, victim and perpetrator, say. Standard analysis, what happens is you basically extract this stain, and this has been set to 50-50, 50-50 mixture here. You basically sample that, and then you, when you sample this, you extract DNA and run a DNA analysis. You obtain an electrophoregram, and you'll have profiles, the alleles that basically have similar peak height, if there's no allele sharing at that point. Whereas the concept we're talking about here is that if one was able to take the mixture itself and sample individual cells from that mixture, right? Because of sampling variation, right? If you were to sample randomly cells from this and do multiple samplings, then what should happen is that you should be able to obtain subsamples of that mixture that would not have the 50-50, that would not all have the 50-50 weight ratio. In fact, we would expect this would be a binomial sampling, because if basically the probability is 50, p is 50, right? So 0.5. What happens is that we would expect a binomial distribution if we were able to remove these cells. Now we can remove these cells because of technology, laser capture technology. Okay, if you to sort of jump to the chase, in this case we're going to be sampling 20 cells, 10 times 20 cells. We're going to capture 20 cells and take 10 samplings. Okay? If you do that, then the binomial distribution is such that approximately 50% of these samples should have the number of cells should range from a 9 to 11, a 10 to 10, or 11 to 9 ratio, which if you're doing a manual deconvolution, it's difficult. But if you're doing it manually, 50% of the time you should have populations that would have 8 and 12, or a 7 and 13, or a 6 and 14. When you have that, you're starting to get a major minor. Okay? So the concept is to basically do subsampling, binomial sampling, and as a result, you should have different weight ratios. Okay, from the same, from a mixture where if you do the standard analysis, you'd have one weight ratio. And that weight ratio is dependent upon what the weight ratio was when you extract the whole lot. So the hypothesis is that multiple samplings of individual cells from mixture will result in an information gain compared to a single sampling for two reasons. One is that it's more likely that a beneficial weight ratio, more informative weight ratio will be obtained. For example, a weight ratio of 0.2 is going to be more informative than a weight ratio of 0.5, right? At least for one of the components. And also the possibility exists of doing a joint likelihood function that's combining data from the multiple samplings, and that will further increase the information. Okay, so what we have to then do for this is to sample individual cells. And we use laser capture and microdissection, which is a technology developed by NIH in 1996 for basically to separate cancer cells from non-cancerous cells. It's gained some interest in the forensic community in the last few years, especially for the separation of sperm and non-sperm cells. The particular laser capture we've used in this work happens to be a Leica. There are different ways of doing laser capture, and as you can see from this, it's basically a microscope with a laser attached to it. In this case, the way we capture, and as I said, there are different ways of doing laser capture with different models. In this case, we're going to basically take some cells here. We use a laser to cut around these cells, and basically the cells fall off by gravity into a tube, in fact, directly into a PCR tube. So this shows, this is a cell, single cell, this is the slide, and we have to place these cells onto a particular proprietary slide with certain chemical composition. Anyway, what basically this shows is a cell, and this is the cap of a PCR tube here, okay? And we can actually check, this is the tube cap, we capture them into the cap of the cells in this particular method, and then we cap the tube. This actually shows you the cells after being captured into the actual cap itself. So we can check that the cells have been taken out, and we can also check that the cells are in the actual tube itself. So what we did here is we prepared some buccal cell suspensions, counted the cells. We then made one-to-one. In this case, we created many mixtures, but of course we can create a one-to-one mixture by cell counting. We apply them to the slide. We don't stain them, we just heat fix. We then look under the microscopes at 400x. We capture the cells, and then in the tube, we then add lysis buffer. Right? We have to burst open the cells directly into the PCR tube. And that's taken some time. The technological advances have been mainly in this lysis approach, okay? So that's what we do, and we then we add a PCR mix, and then we just do PCR amplification. So how good is this system, right? So I just want to give you some basic data as to how efficient this system is before we talk about the mixtures. And basically, I'll show you data from 10 donors, 5 female, 5 male, and we've collected 1 cell samples, 2 cell samples, 3 cell samples, 4, 5, 10, and 20. We've done 10 replicates, both biological replicates and technical replicates. And then we amplify, in this case, with 34 cycles. So this is an LCN technique, using LCN now as a technology. And we separate using a standard capillary electrophoresis system. And for the aficionados in the room, it's the identifier system, which is a multiplex system used in casework widely. So if you look at the success rate, if you take the number of microdissected cells, if you take 20 cells, and we've done multiple cell samplings, 99% of the time we will get a partial profile, 99% of the time. In fact, 80% of the time we'll get a full profile with 20 cells. So even with one cell, 74% of the time we will get a partial profile. Only 4% would be a full profile, and but there's 26% no profile. So we've used 20 cells in this particular study here. The results I'm going to show, but the work is underway to improve profile recovery from a single cell. Because obviously, if you can get a profile from a single cell, we don't have to get involved in mixture deconvolution. Now that will still raise. We still need interpretation for single cells, but that's another day and another story. But so in this case, the actual data we're going to show here is we created two mixtures, two 50-50 mixture samples, male-female and a female-female, and we've taken 20 cell samples from each mixture, and we've done 10 separate samplings. So there's 20 cells collected 10 times, okay? And so they're collected randomly from the slide. The slide has been created with a mixture, a one-to-one mixture, and we've just randomly selected the cells. We then obtain a DNA profile from this, and then we perform quantitative computer interpretation of the data in this case. And this is just to show what a profile would look like from an example of a 20-cell, one of the 20-cell samples with a one-to-one mixture. And you can see that at all loci there are alleles. That's one would hope. And in fact, if you look at a single channel across multiple cell samplings, you can see that the weight ratio, you probably can't see it clearly in this slide, but basically here are 10 different samplings, and the weight ratio is given by the omega. That's 21% weight ratio, 57, 54, 20, 39, 20, etc., etc. So the weight ratio does change. And with that, I'd like to hand it over to my colleague to finish the talk.
Thank you. So I'll talk about quantitative computer interpretation. And the goal is to answer these questions. Does binomial sampling diverge from 50-50? Does this increase identification information? And how else can we increase information yield? And the approach is to take some information solution that can infer mixture weights, that can infer genotypes, can compute log LR match information, and of course represent uncertainty using probability. The approach we're taking is a trivial casework system, which you can read about in a forthcoming paper. It's on our website. You can download that. And the main points here is that we have a hierarchical probability model that accounts for all relevant variables in the experiment: prefamp, stutter, degradation, data combination, peak uncertainty, and so on, handling 3, 4, 5, 6 contributors, and of course modeling genotype and mixture weights. It runs Markov Chain Monte Carlo statistical search over the 1,000 or so variables, and it's been extensively validated, courtroom proven, and government approved at this point, and it's been used on about 100,000 samples so far, involving rape, murder, burglary, kinship, mass disaster databases, and so on. So this is what the mixture data look like. You say here are 3 peaks, and this comes from the 2-contributor 50-50 ratio binomial sampling of a laser capture microscope cells with the 34 cycles. So a way to visualize the likelihood function, because that's at the heart of what all of us do here, is imagine here's one experiment, D16, of one of the different samples, and in green you see the data. And so you can hypothesize that maybe there's one individual who's a 12-13, another individual who's an 11, and here's the first contributor who's homozygote 11, and a heterozygote individual, and you have a certain amount of DNA, and you put it there. Those are 2 of the main variables, the 2 allele pairs, as well as the relative amounts. And now if you were to expand that, accounting for the second-order effects, the experimental variables, stutter, and so on, you see in gray that there's a pattern that's a model that's trying to account for the data shown in green. And a likelihood function is a mathematical way of explaining that pattern. A better explanation gives a higher probability. Explanations that have nothing to do with the data have a 0 probability, and things in between give you something in between. And what a computer can do is to try out, by statistically, every possible combination that could exist, hundreds of thousands of things that a person generally doesn't do in the day or two they have allocated to review a case. So that's the likelihood function and the complete search of it. So one of the random variables is the mixture weight. This is what a Markov chain looks like for the first contributor, for how much there is of the template, about 25%, and for the second contributor shown in orange. And if you take the histograms, which the computer does, from those Markov chains, you get reasonably normal-looking distributions showing 25 and 75%. Genotypes are also a random variable. Here, the second contributor has about 100% probability at that locus for the second contributor. And for the first contributor, there's some uncertainty, and that genotype uncertainty is described by allele pair probability of these two different allele pairs, 1111 and 1112. One's more probable than the other. The probabilities add up to 1. And an important thing to state here is that at this point in the computation, the system has never seen any reference sample, any suspect. So this is a guaranteed objective way of inferring a genotype, which can be helpful in court because it is completely objective. So once we have our posterior probability, here the same one I've just rotated at 90 degrees, shown in blue, we can form a likelihood ratio. How do you do that? Well, somebody presents you with a suspect, and we don't know what that suspect is ahead of time, but here's an allele pair, which happens to be 1111. And in brown, you can imagine across the whole room, there's a probability distribution of little brown bars. That's the prior population of this distribution of the population, about 100 allele pairs. And so if following S and Muller from 1938, if you take the ratio of the posterior probability to the prior probability, you get some number. In this case, it's about 8. And that tells us how much more the suspect matches the evidence than a random person. There's a description of many ways of writing down the likelihood ratio in usual form, starting with Jack Good's Bayes factor and ending up with these ratios of probabilities that you can read about. And that ratio gives you a likelihood ratio. And when you take a number like 8 or 10 and multiply 15 different loci, you end up with a joint likelihood ratio that's joint across all the loci. Here it's shown as the logarithm, as 10, 100 on a logarithmic scale. And when you look at the different likelihood ratios for the first contributor or for the second contributor, I think that's the minor, that's the major, you pretty much recover the information. This is looking at one of the samples from the low copy number data, 10 to the 16th, 10 to the 18th. So to answer the first question that we asked, does binomial sampling actually produce some deviation from 50-50 to give potentially more informative profiles? Well, this is the distribution of the roughly 40 matches. As Jack said, there were 10 samplings that were male-female pairs, 10 samplings that were male pairings. I think it was actually female-female, male-female. And then relative to each of the two individuals, there's a match. So that's how you get the 40. And if you look at a histogram of what those matches are, they're not clustered all at 50-50, but they are spreading out into mixed proportions ranging from 20 to 80%. So the first answer is yes. Jack's binomial sampling seems to be working well. Now, if we take those roughly 40 matches and we sort them by mixture weight, ranging from 20% up to 80%, and we ask, how much information is there? What's the log likelihood ratio on a scale of 0 to 20? We see that with just one sample, there is quite a bit of information, and the ones down at 20% are informative and the ones up at 80% are informative. So that separation away from 50-50, to explain the data when the peak heights are different can be more informative than when they're all collapsed and they look equivalent. So we can also ask, how do you get more information out of the system? And for that, you can use more than one sample. Conceptually, suppose you had 5 different samples. I'm just looking at one locus. You can have a joint likelihood function that hypothesizes a model, in this case, of genotype allele pair values. And now it has to explain all of that data. When you have more data in statistics, there's more constraints on what the answer can be, and you can extract more information. So here's the hypothesis. Here are what the patterns would look like. And so we did that on this LCN data, 34 cycles, just looking at all possible pairings. So these are just joint likelihoods with 2. And for each of these samples, we formed all possible pairs. And what you're seeing is the maximum of the 9 pairings. At the top, what was the total information? And in red, you're seeing the information gained. So by doing a joint likelihood function on just 2 samples, you're basically achieving full information of 10 to the 18th or so across the whole population of allele pairs, showing that random sampling works well and joint likelihood functions work well as well. And since I have at least 3 or 4 minutes left, I'll show you some low-level STR data that was 28 cycles, where we have delightful peak heights of 20, 30, 40, and so on. And looking at D16 this way, I just wanted to show you. So here's a nice calculation where we combined all 10 samples, and we have the Markov chains here for all 10. And here are the mixture weights, which you see there's some around the center, some splitting. You can imagine that that's sort of a nice binomial distribution. And the recovery here is 100% probability at every allele pair. That's only one allele pair that's appearing in the probability. And when you take a look at the information, the match information, the log likelihood ratios, you're getting that 10 or 100 again, and you completely recover the profile by looking at a joint likelihood function with all 10. That's mainly for fun. So observations are that binomial sampling does indeed separate DNA mixtures. Probabilistic genotypes do extract considerable information. The true allele system does quantify mixtures and information, and so it's helpful in doing these studies. And statistical data, as well as statistical interpretation, are useful. And we'd like to thank the people who helped out on the study, generating data and so on. And in the 30 seconds left, I'd be happy to entertain any questions with Jack. Thank you.